Have you ever tried carrying someone else's baby, only for the baby to end up crying? Why do babies cry? How do mothers feel when babies cry? Infant cry actually plays an important role. The cry helps to maintain proximity with their mothers and signal for caregiving. Mothers' psychobiological mechanisms have evolved over the years to respond to infant cries as they experience increased stress from higher autonomic arousal. What has been studied so far? Paul and Mosbach, Seifrieds, Swain, and Pialini's respective teams have studied the stress response in mothers towards infant cry through electrophysiological methods, heart dynamics, and general brain responses in mothers through fMRI. Important points include how mothers exposed to infant cries experience more stress and were unable to concentrate on non-infant care tasks, and that the anterior cingulate cortex is deactivated in females during infant cry. McEwen and Gianaros have also discovered that the hippocampus, amygdala, and prefrontal cortex are involved in stress responses. Do mothers then feel stress when hearing an infant cry relative to non-mothers? Through this proposed study, we will understand the differences in neural stress responses between mothers and non-mothers. This will provide insight on how mothers' brains are attuned differently for infant caregiving. Based on previous research, we hypothesized that mothers would experience more stress than non-mothers in the infant cry stimulus compared to the white noise stimulus observed by mothers' higher neuroactivity in the hippocampus, amygdala, and prefrontal cortex, and both mothers and non-mothers would experience a deactivation of the anterior cingulate cortex towards infant cry stimulus. To test this hypothesis, we will recruit 25 mothers and 25 non-mothers to undergo fMRI scanning. All participants will listen to an infant cry and a white noise stimulus through earphones in the fMRI scanner. The stimuli will be presented using a block design with 2 runs of the stimuli task containing 12 blocks in each run. Each block will contain either an infant cry stimulus or a white noise stimulus that will last 20 seconds each, and a 20-second rest between each stimulus block. To analyze the data collected, we will use 2 levels of analysis. A time series analysis is first used to test for differences at the individual level, followed by a region of interest analysis to test for differences between mothers and non-mothers. We predict significant activations in the hippocampus, amygdala, and prefrontal cortex towards infant cry for mothers compared to non-mothers, with no significant activation in those areas towards white noise for both groups. Observing these activations means that brain regions involved in stress response are activated during an infant cry, and this effect is only seen in mothers and not non-mothers. This has health implications for mothers who face stress often due to the constant cries of infants. If no significant activation is observed, this implies that infant cry may not induce stress responses in females regardless of parental status. This may mean that mothers may have an adapted stress response to infant cries, such that mothers do not feel stress with frequent exposure to infant cries, although their physiological responses show otherwise. We also predict that both mothers and non-mothers would show significant deactivations in the anterior cingulate cortex when presented with infant cry. If there is significant deactivation in the anterior cingulate cortex, it would suggest that infant cries can reduce cognitive functioning as the anterior cingulate cortex is involved in functions like attention allocation. Future studies may incorporate a whole brain analysis on stress responses and explore the role of the HPA axis on stress responses across different population groups, such as gender and the number of children. Knowing the mechanisms behind infant cry and the mother's stress response towards it will improve our understanding of the mother-infant relationship and the well-being of mothers.